Picture this. You're looking through a microscope at a brain cell taken from someone with late-stage Alzheimer's. Most of its neighbors are gasping for life, or already gone. But this one? It's cool, calm, business as usual. Today we're going to slip inside that survivor cell and ask a simple question. How on earth is it still standing? Here's the headline. The cell isn't lucky, it's prepared. While surrounding neurons clog up with toxic proteins, this one is running a cleaner, faster housekeeping routine, spotting trouble early and tossing it out before it spreads. Think of two houses on a flood-prone street. One owner stacks sandbags and clears the drains long before the rain hits. Same storm, very different outcome. First signpost. How did scientists even notice these outliers? Instead of the usual healthy versus diseased comparison, researchers zoomed way in. They examined single neurons that sit side by side in the same damaged tissue but live wildly different fates. That tight contrast let them search for molecular habits, tiny routines that separate strugglers from survivors. One habit jumped off the page. Second signpost. Meet the cleanup crew. Two workhorse molecules called BAG3 and HSP70. They're chaperone proteins, normally busy folding other proteins the right way. In these hardy neurons, they're doing extra credit, teaming up to catch tau fragments right before those fragments turn rogue. Here's how the play unfolds. BAG3 and HSP70 tag the sketchy tau pieces. Then, a special form of autophagy, cellular recycling, pulls only the bad parts off the shelf and feeds them to the lysosome, the cell's shredder. No scorched earth, just a surgical recall. In survivor cells, this loop is running hotter, faster, louder. And the wiring of those neurons stays intact even in late-stage tissue. Now, scientists love a good test. So they dialed that loop up in mouse models engineered to pile up human-style tau. They simply boosted BAG3 levels in vulnerable brain regions. Result? Tau buildup plunged. Even better, the mice ran mazes with fewer wrong turns. Translation: protect the circuit, protect the function. But every good story needs a villain. Enter calpain-2, a protease that revs up in diseased brains. Calpain-2 chops tau into pieces that slip past ordinary controls and clump together more easily. Wherever calpain-2 spikes, the BAG3 HSP70 defense struggles to keep up. It's a molecular tug-of-war inside stressed neurons. Could we nudge the good guys instead of only fighting the bad? Researchers tried it in a dish using the sugar trehalose, known to boost autophagy. Trehalose bumped BAG3 levels, hinting that cells can be coaxed to lean harder on this defense. But there's a Goldilocks zone. Too little, no real effect. Too much, and you start sweeping up parts the cell actually needs. Let that sink in for a second. Third signpost. This isn't a simple on-off switch. It's more like a soundboard with multiple sliders. One slider flags risky tau fragments. Another routes cargo to lysosomes. A third tones down calpain-2 before it floods the system with trouble. Different neuron types set those sliders differently, which might explain why memory circuits fail early while motor or sensory areas hang on. That idea reshapes clinical trials. Instead of only asking, did plaques or tangles shrink? Future studies may track how well this internal circuit stays tuned over time, and whether people with naturally higher BAG3 settings decline more slowly even when classic pathology is present. Looking ahead, scientists want to build resilience signatures. Imagine a brain checkup that scores how active your BAG3-linked circuit is, the way a stress test reveals hidden strength in a bridge. Lifestyle tweaks, better sleep, regular exercise, maybe diet, could become daily ways to nudge those sliders into the safety zone. Prevention might end up looking less like one magic pill and more like tuning an orchestra of protective pathways. So here's what we covered today. 1. Some neurons survive Alzheimer's damage by cranking up a housekeeping loop powered by BAG3 and HSP70. 2. Dialing that loop higher in mice slashed toxic tau and kept their memories sharp. 3. The villain calpain-2 can tilt the balance the other way, but targeted nudges, like trehalose, show the circuit is adjustable. And 4. The future of Alzheimer's care may shift from clearing debris to boosting each brain's built-in resilience. The empowering part? Many of those sliders might be tweakable. By drugs, yes, but also by how we sleep, move, and eat. The more we learn, the more it looks like we're not helpless bystanders. We can help. Train our brains to spot storms early and reroute around them. Thanks for listening. If today's episode lit a spark, share it with a friend and make sure to hit subscribe. Until next time, keep that brain of yours resilient.